Hello, my name is Alex Weigman, and today we'll be looking at the eye. No pun intended. The eye is a special sense, part of the nervous system, and is very complex. We'll be talking about the detailed anatomy, the detailed physiology, and the common pathologies of the eye. So let's dive right in. The eye is located within the orbit of the skull, which is made up of 7 bones and broken up into 4 different sections. There's the roof, which is made of the frontal bone, a small bit of the sphenoid bone, and separates the contents of the brain and the orbit. There's also the medial walls, which are made of the maxilla bone, the lacrimal bone, a greater portion of the ethmoid bone, and the sphenoid bone. There's also the floor, which is made up of the maxilla bone and a small bit of the zygomatic and palatine bones. And finally, there's the lateral wall, which is made up of the zygomatic bone and the greater wing of the sphenoid bone. There are 5 fissures and foramina within the orbit of the skull that allow various vessels to pass through. There's the optic canal, which allows the optic nerve and the ophthalmic artery to pass through. There's the superior orbital fissure, which allows the superior and inferior branches of the ocular motor nerve, trochlear nerve, abducent nerve, the lacrimal, frontal, and nasociliary branches of the ophthalmic nerve, and the superior ophthalmic vein to pass through. There is the inferior orbital fissure, which allows the maxillary nerve and its zygomatic branches, the infraorbital vessels, and the vein communicating with the pterygoid plexus of the veins to pass through. The infraorbital foramen, which allows part of the maxillary nerve to pass through. And there is the anterior and posterior ethmoidal foramina, which allow the anterior and posterior ethmoidal nerves and vessels to pass through. We will now look briefly at the muscles of the eye. There are 2 types of muscle groups. There's the extrinsic, which is responsible for the movement of the eyeball and the raising of the upper eyelids, and the intrinsic, which controls the shape of the lens and the size of the pupil. Of the extrinsic, there is the levator palpebrae superioris, the superior rectus, which is responsible for elevation, adduction, and medial rotation of the eyeball, which is innervated by the superior branch of ocular motor nerve, or cranial nerve 3. There's the inferior rectus, which is responsible for the depression, adduction, and lateral rotation of the eyeball, which is innervated by the inferior branch of the ocular motor nerve. There's the medial rectus, which is responsible for the adduction of the eyeball and is innervated by the inferior branch of the ocular motor nerve. There's the lateral rectus, which controls the abduction of the eyeball and is innervated by the abducent nerve. There's the superior oblique, which is responsible for the depression, abduction, and medial rotation of the eyeball, which is controlled by the trochlear nerve. And finally, there's the inferior oblique, which is controlled by the inferior branch of the ocular motor nerve and is responsible for the elevation, abduction, and lateral rotation of the eyeball. Of the intrinsic muscle group, there's the ciliary muscle, the sphincter pupillae, and the dilator pupillae, which we will talk about later. We will now look at the vessels of the eye and its associated structures. Of the arteries, the main artery is the ophthalmic artery, which branches off to become the lacrimal artery, the central retinal artery, the long and short posterior ciliary arteries, the muscular arteries, the supraorbital artery, the posterior ethmoidal artery, the anterior ethmoidal artery, the medial palpebral arteries, the dorsal nasal artery, and the supratrochlear artery. Among these, the long posterior ciliary, the short posterior ciliary, and the anterior ciliary arteries feed the eyeball itself. As for veins, there are the superior ophthalmic vein and the inferior ophthalmic vein. And as for innervation of the nerves, there are many nerves that feed the eye. There's the optic nerve, the oculomotor nerve, the trochlear nerve, postganglionic sympathetic fibers, the ophthalmic nerve, the lacrimal nerve, the frontal nerve, the supratrochlear nerve, the supraorbital nerve, the nasociliary nerve, the long ciliary nerves, the posterior ethmoidal nerve, the infratrochlear nerve, the anterior ethmoidal nerve, the ciliary ganglion, the parasympathetic root, the sensory root, and the sympathetic root. Reflecting on the general structure of the eyeball itself is a round structure with a posterior projection and is located in the anterior portion of the orbit. There are 2 chambers in the anterior portion of the eye, the anterior chamber, which is right behind the cornea but in front of the iris, and the posterior chamber, which is behind the iris but anterior to the lens. These 2 chambers are continuous and filled with aqueous humor. This aqueous humor is secreted in the posterior chamber and flows through the pupil into the anterior chamber, which is absorbed by the sclerovenous sinus. The humor provides nutrients to the cornea and lens, which are avascular, and the circulation also causes a constant intraocular pressure. The lens separates the anterior 1/5 of the eye from the posterior 4/5. It is a biconvex disc which is attached to ciliary bodies by suspensory ligaments. The anterior surface is made up of cuboidal epithelial cells, while the posterior is made up of long columnar epithelial cells, which are called lens fibers. These lens fibers lose their organelles and nuclei and become an accumulation of proteins called crystallines. These crystallines are covered by a capsule and then make up the lens. Finally, there's the postremal chamber, which contains vitreous humor. The wall of the eye is composed of three layers: the outer fibrous layer, the middle vascular layer, and the inner layer. The fibrous layer is composed of two parts, the sclera and the cornea. The sclera is made up of opaque, dense connective tissue. It is also called the white of the eye. It is important because muscles attach to the sclera and vessels innervate through it. The cornea is the second part of the fibrous layer. It is transparent, composed of collagens, proteoglycans, and elastic fibers. It is continuous with the sclera. It allows for light to enter the eye. The central part of the cornea receives oxygen from the outside air. And in 2013, a research group from the University of Nottingham published a paper stating that they found a new layer in the eye called Dua's layer. Right now, Dua's layer is controversial, as they are not sure if it's part of another membrane of the cornea or if it is a completely new layer, and they have to revamp textbooks afterwards. The vascular layer contains three parts: the choroid, which is 2/3 of the vascular layer. It is thin and highly vascular and strongly attached to the retina while loosely attached to the sclera. The ciliary body, which is the anterior border of the choroid, contains two parts: the ciliary muscle, which is smooth muscle under parasympathetic control of cranial nerve 3, and the contraction causes a decrease in the size of the ring formed by the ciliary body. The reduction in tension on the suspensory ligaments causes the lens to become more round. The second part of the ciliary body is the ciliary processes, which are ridges from the inner surface of the ciliary body, and zonular fibers attach to the processes, which are then attached to the lens. The final part of the vascular layer is the iris, which is the colored part of the eye. It contains muscles that control the size of the pupil. These are the intrinsic muscles that we talked about before: the sphincter pupillae muscle, which is under parasympathetic control, causing a decrease in pupillary opening when stimulated, and the dilator pupillae muscle, which is under sympathetic control, which causes an increase in pupillary opening when stimulated, which makes sense. When you are in a fight or flight situation and your sympathetic nervous system is kicking on, you want to take in as much light through your eye to see as much as you can in any threat. Therefore, your pupils become large when you are in a fight or flight situation. And as I said, the iris is the colored part of the eye, and the color is dictated by the amount of melanin within the iris. So the more melanin you have, the darker your eye. So brown eyes contain the most melanin, and blue eyes contain the least melanin, and they are the lightest in color. But the genetics behind figuring out eye color is kind of complex. It has to do with chromosome 15 and 17 and other genes in between. Finally, there's the inner layer of the eye. The posterior aspect of the inner layer has the optic part of the retina, while the anterior portion of the inner layer is non-visual. The junction between these two portions of the eye is called the ora serrata. So we'll look quickly at the optic part. There is a pigmented layer, which is the outer layer, which attaches to the choroid layer and continues over the internal surface of the ciliary body and iris. There is the neural layer, which is the inner layer, which only attaches to the pigmented layer around the optic nerve and at the ora serrata. And the neural layer contains 6 to 7 million cones and about 120 million rods. Finally, there's the posterior surface of the retina, which contains the optic disc. This is where the optic nerve leaves the retina, where there are no cones or rods. It's a blind spot. The macula lutea is lateral of the optic disc, and the fovea centralis is in the center depression of the macula lutea. In the fovea centralis, there's a high concentration of cones, very few rods. This is where there's the highest visual sensitivity, and light is concentrated the most there when looking at an object. So now that we looked at the detailed anatomy, we're going to start looking at the detailed physiology. The physiology of the eye is very complex. There's a lot of molecular and cellular biology as well as biochemistry. These are my forte and what I take a lot of interest in, so I hope that you find this as interesting as I did. So a brief overview of how the eye works. the eye takes in light through the pupil at wavelengths of 380 nanometers to 750 nanometers, which is considered visible light. Light rays will diverge due to refraction as it hits the lens at the thin center, while they converge at where the lens is the thickest. Converging light rays come together at a focal point, forming an inverted image, and this inverted image is then flipped right side up in the visual cortex of the brain. The lens will change its shape to create a more focused image with the muscles that I listed in the anatomy section. So we're going to look at a detailed view now. And how does the eye react to light? It's all in the retina. We're looking at that neural layer, also called the photoreceptor layer, which contains the 6 to 7 million cones and 120 million rods. So we're going to look at both cones and rods, but we're going to look at rods first. So rods are responsible for non-color vision. They contain about 700 double-layered membranous discs that contain rhodopsin. What is rhodopsin? This is rhodopsin. Rhodopsin is made up of a GPCR protein known as opsin and 11-cis retinal. So 11-cis retinal attaches to the lysine residue 296 to form a shift base, which allows for absorption of longer wavelengths than retinal by itself. So retinal by itself will absorb wavelengths up to 380 nanometers, while the shift base can absorb wavelengths up to 440 nanometers. It's also believed that the shift base is protonated due to the fact that the rhodopsin can absorb up to 500 nanometers. Now we're going to look at the progress of how light reacts with rods. So a photon enters and converts 11-cis retinal into 11-trans retinal, causing opsin to change into its activated state called bathrhodopsin, which is all trans and a protonated shift base, and then within milliseconds becomes metarhodopsin 2, which is an all trans, but the shift base is deprotonated, which then interacts and activates G-alpha T. We're going to set G-alpha T aside real quick, and we're going to look at what happens to opsin. So activated opsin itself is unstable and breaks down at opsin into trans 11 retinal, which ends visual signaling. And then the trans becomes cis again in the dark and binds with opsin, which forms rhodopsin, and the cycle continues. Going back to G-alpha T. G-alpha T, when activated, triggers an exchange of GDP for GTP. GTP then binds the beta-gamma subunits of transducin and are released, which then makes the alpha subunit turn on phosphodiesterase. Phosphodiesterase then turns cyclic GMP into GMP through hydrolysis. So right now we're going to take a quick break and look at the rod cells and how they are in the dark and in their natural state, and then we're going to tie it all together in the end and see how neural signaling works. So in the dark, the membrane potential of the rod cell is usually around negative 30 millivolts, which causes them to secrete neurotransmitter due to being in a constant state of depolarization. This is caused by a large number of open sodium, calcium, and potassium ion channels. When light comes in, it causes the channels to shut and the membrane potential to become even more negative, and we perceive the lower amount of neurotransmitter released as light. So tying everything together from the beginning to now, the channels are controlled by cyclic GMP, which is a secondary messenger. There's usually a very high concentration of cGMP outside of the rod cell. cGMP-gated cation channels stay open due to an increased level or concentration of cGMP. cGMP goes down in light due to phosphodiesterase converting cGMP into GMP, which then in turn causes ion gates to shut and the membrane potential to hyperpolarize. This all in turn lowers the amount of neurotransmitter released and causes neural signaling. So we're going to look briefly at cones. Cones are found within the central fovea. They work very similar to rods. There are 3 distinct photoreceptor proteins in the human eye. There's S, which stands for short, picks up wavelengths of 426 nanometers and interpret the color blue. There's M, which stands for medium, which pick up wavelengths of 530 nanometers and interpret the color green. And L for long, which pick up wavelengths at 560 nanometers, which is interpreted as red. Some interesting things are cones are about 40% identical in sequence to rhodopsin. Blue is about 40% the same as green and red in sequence, while green and red are more than 95% the same in sequence to each other. We're now going to look at the common pathologies of the eye, starting with cataracts. I look at Matthew 6:22 through 23, which says, "The light of the body is the eye. If therefore thine eye be single, thy whole body shall be full of light. But if thine eye be evil, thy whole body shall be full of darkness. If therefore the light that is in thee be darkness, how great is that darkness?" Not saying that people with cataracts are evil, but it does relate with how cataracts works. The pathology behind cataracts is it's a clouding of the lens. It's caused by advanced aging, infection, or trauma. It is the leading cause of blindness worldwide and it can occur at any age. Cataracts can be removed through surgery. They'll replace your lens, or they can give you glasses that'll help out with that. Our next pathology is glaucoma, which is damage to the optic nerve, which leads to vision loss and blindness. It's actually a group of diseases. The pathophysiology is when normal fluid pressure in the eye slowly rises, and there are two types of glaucoma. There's open angle, which is chronic, and closed angle, which is sudden and painful. Treatments behind glaucoma is surgery, laser surgery, and medications. Macular degeneration is the degeneration of the macula causing impairment of sharpened central vision, although peripheral vision is maintained. There are two types of macular degeneration, wet and dry. Wet is caused by the growth of abnormal blood vessels behind the retina that leak blood and fluid, causing scarring and rapid loss of central vision. An early sign of wet macular degeneration is straight lines appear wavy. Then there is dry macular degeneration, when the macula thins over time and blurs central vision. This progresses slowly. An early sign is drusen, which is a yellow or white deposit under the retina. It usually affects both eyes. Treatment includes, for wet macular degeneration, various medications, and there is no FDA-approved treatment for dry macular degeneration. A change in diet and nutrition also helps as well. Diabetic retinopathy is a complication of diabetes and is the leading cause of blindness in the United States. It causes damage to vessels supplying blood to the retina and involves optic nerve degeneration, cataracts, and retinal detachment. There are 4 stages to diabetic retinopathy: mild non-proliferative retinopathy, moderate non-proliferative retinopathy, severe non-proliferative retinopathy, and proliferative retinopathy. This disease usually affects both eyes. Treatment includes controlling blood sugar and blood pressure, as well as lipid levels, surgery, laser treatment, which seals off bleeding, and various medications. Finally, there are refractive errors, which include myopia, which is also called nearsightedness. This is when one can see close objects clearly, but distant objects are blurry. It's when the focusing system is too powerful or the eyeball is too long, also called axial myopia. It's corrected by concave lenses that counter the refractive power of the eye. It can also be corrected with corrective eye surgery, radial chorotomy, or LASIK. There's also hyperopia, which is also called farsightedness. It's the case when distant objects are clear, but close objects are blurry. The cornea and the lens system are optically too weak or the eyeball is too short. The image can also be focused behind the retina. Treatment includes convex lenses, which cause light rays to converge. There's presbyopia, which is unavoidable degeneration of the accommodation power of the eye. It's associated with age. The lens becomes sclerotic and less flexible, and the treatment is usually reading glasses. Finally, there's astigmatism, which is a refractive error in which affects the quality of focus. The corneal lens is not uniformly curved, thus light rays do not focus on a single point. There are 2 types of astigmatism. There's regular astigmatism, which is corrected by glasses formed with the opposite curvature gradation, and there's irregular astigmatism, which is an abnormal form of the cornea, fits no specific pattern, and is very difficult to correct with glasses. So there you have it, the human eye, an amazing and complex gift that God gave us so we can take in His glories and the amazing visual gifts He gives us daily. I hope you enjoyed this video, and God bless.